Hello and welcome to your lesson on cell division. So this is the first topic in the last unit of National 5, and the unit is called multicellular organisms. So this unit is all about kind of moving further up from just one cell into organisms that are made up of lots of cells and looking at some of the processes that are involved in keeping them alive. So we're looking at things like plants and animals and some of their systems and processes. So the first booklet in this unit is topic 1, and it is called Producing New Cells, and it kind of directly follows on from the last unit. We're going to look at how cells divide and how they then form more complex organisms that are made up of so many different specialized cells. So some things perhaps to think about here, some questions just to kind of get us started. We're going to be thinking about 3 reasons for cell division. So why do cells have to divide? We're going to need to refer back to something called the chromosome complement. We learned a little bit about that in the last unit when we looked at DNA. We're going to look at what the chromosome complement is in humans. And we're going to be thinking about the number of sets of chromosomes in a haploid cell and a diploid cell. So a lot of this is just revision of stuff that we did in that DNA topic in the last unit. Now, I'm not going to give you answers to those straight away. Feel free to pause the video, write down what you think the answers to them might be, and then we will kind of reveal these answers as we go through today's lesson. Okay, so the first thing really is, why is it important for the nucleus to divide in 2 before the cell divides? So you can see a picture here. This is actually an amoeba, but you can see that this amoeba is going to divide. But you'll notice that the first thing that has to happen here is that the nucleus has to divide. So this cell briefly has 2 nuclei, and then eventually the rest of the cell divides. So the cytoplasm splits, the cell membrane splits, and we now have 2, what we sometimes call daughter cells, but these came from that. So they are pretty much genetically, they're identical to that. So why is it important for the nucleus to divide in 2 before the cell divides? So think about what is in the nucleus. What is it that's found there that these 2 daughter cells would need to be able to survive? Well, the answer is that the nucleus contains the DNA that controls the activities of the cell. So a new cell would need the information in the nucleus to be able to control itself. So that's what we're needing to pass on. We're needing to pass on the DNA, the instructions for the cell to be able to control itself. Now, the chromosome complement, something we've heard before, the chromosome complement of a species is the number of chromosomes that the cell requires in order to function properly. So we're really referring to the diploid cells here. So in a diploid cell, in a diploid human cell, we would have 46 chromosomes in the chromosome complement. You can see them all laid out here in that picture. But for different species, that complement might be slightly different. So for a roundworm here, it's only 4, rice plants, 24, chimpanzees, 48, dogs, 78. So the complement is the number of chromosomes that the cell requires to function properly for that specific species. Okay. And when cells divide, that chromosome complement must be maintained. So if we've got a human cell that's going to divide and it's got 46 chromosomes in it, then when it divides, the 2 daughter cells that come from it are also going to need those 46. And when they divide, they also need that 46, and so on and so on. So after 3 divisions, we now have 8 cells along the bottom that all have the exact same genetic information, and that is crucial for the cell to be able to function. So why is cell division important? Well, have a look at this picture, and I want you to try and identify the haploid cells that are in the diagram, and then the diploid cell that's in the diagram. Okay, so the haploid cells in this diagram are the egg and sperm. They've got the 23 chromosomes. And remember, they're going to join together at fertilization, and they're going to produce a zygote that is now going to have 46 chromosomes. And that is the 23 plus 23. So these 2 cells here are the haploid cells. The zygote is the diploid cell. And every cell in this embryo will also be diploid. It will have the 46 chromosomes. So where in the diagram is cell division important? Well, the important bit here, so there's no cell division going on here. This is fertilization. But once we've got the zygote, it then has to divide again and again and again and again, so that we get loads and loads of cells that form the embryo, and they've all got the same 46 chromosomes. So you can see that broken down a little bit more here. So this is our fertilized egg or zygote, and it then has to divide, and again, and again, and again, and again. And it's getting more complex. So you'll see, obviously, we've gone from just a ball of cells to something a lot more complicated by the time we get to this. So there is another process that goes on. Some of these cells are becoming specialized. We'll talk more about that in the next lesson. But what is important is that even though they may be becoming specialized, to actually just get to something this big, we have had a whole lot of cell divisions going on. And in every single case, we've got the 46 chromosomes going into each and every one of those cells. So mitosis and cell division are essential for growth. Cell division and mitosis, sometimes these words are a bit interchangeable. So when a cell divides, it is splitting into two. But mitosis refers in a bit more detail about what's going on in the nucleus and with the chromosomes. So we're going to be focusing on this during this lesson. Now, the reason why cells need to be able to divide, well, we can see one reason here, that is growth. We are going from one single cell to lots and lots and lots of cells. And even after birth, growth is still continuing. And as we grow, it's because our cells are dividing. So we're increasing the number of cells in the organism. But there's a few other reasons why cells need to be able to divide. So the first one is for repair. At lots of times in our life, we're going to injure ourselves, we're going to get damage, and when that happens, these cells need to be able to repair and replace the damaged parts, and that involves more cell division. So it's very localized cell division. It's only happening where the injury is, but it's the exact same process as that's involved for growth. The other reason why cells need to be able to divide is that for some cells, for unicellular organisms that are only made of one cell, when they divide, that is effectively reproduction. Okay, so this is it's just like the amoeba I showed you earlier. The amoeba has doubled its nucleus, and that's divided, and then the rest of the cell divides, and we now have two daughter cells that are identical to the original. So this is a form of reproduction for unicellular organisms. That's things like bacteria and yeast. So the question that we need to be asking, have a look at this. So we've got 46 chromosomes in this original cell. But when we divide 46 by 2, we would get 2 lots of 23. So that's a bit weird. So how do we go from 46 to 2 more lots of 46 from this one original cell? And that leads into the stages of mitosis. So inside the nucleus of every cell, you have DNA stored as chromosomes. So we've got our 46 chromosomes in a human cell, that's 23 pairs. But when our cell divides, we want 46, just like this, in the 2 cells that are going to come from it. So something must happen, because if we just split 46 into 2 lots of 23, we would not get the 46 that we need in each of those daughter cells. Normally, in your cell, you've got 46 chromosomes that look a little bit like this. So it's got all the genes on it, like that, but this is our individual chromosome. Now, at the moment, we can't divide this chromosome. If we just cut this chromosome in half, we're going to have problems, because half would go to one cell, half would go to the other cell, but that means that some genes would be missing in each cell, so that doesn't work. So before we can divide our cell, the chromosomes are going to have to do a little duplication. And you can see we've got 6 stages in mitosis, And if that is the nucleus of the cell before it starts to divide, it needs to do this. And you'll see that the chromosomes in this stage look slightly different. Okay, so something's happening between stages 1 and 2. And this is what it is here. So the chromosome has to make an exact copy of itself, and that's a process called DNA replication. And you'll learn more about that if you take higher. And when this chromosome duplicates itself, it looks like this. So a chromosome that has been duplicated, we say that it is made up of 2 chromatids. Okay, so this is a chromosome, and it's only got 1 copy, so it's got 1 chromatid. This is that chromosome after DNA replication, and it's duplicated itself. And we say that this is a chromosome that has 2 chromatids, kind of 2 copies of itself. Okay, and that's really important. So what we're going to look at now is we're going to look at all the stages of mitosis. So we're going to break it down into 6 stages. Now, in your notes, what you'll have to do is you are going to have to explain what's happening in each of these stages, but you've already got the diagrams there. They're exactly the same as these ones that I'm showing you just now. So, in the first stage of mitosis, what's going on in the nucleus? Well, at this point, the nucleus contains long, uncoiled chromosomes. It's a bit untidy, okay? They've not tidied themselves up into the nice shapes that we need them to be in. So in a way, it's probably a little bit like your bedroom. It's organized chaos, okay? So we need to tidy that up. But the other thing that we're going to need to do is we are going to need to make sure that all of the chromosomes have the 2 copies, the 2 chromatids that we're going to need for cell division. So the cell will be spending a bit of time doing this DNA replication to make sure that when it tidies up this DNA into the chromosomes, that the chromosomes have got the 2 chromatids, the 2 copies, so that we're good to go. So this is stage 1. In stage 2, you'll see that the nucleus is now a little bit tidier, and the DNA has been put back into a nice tidy format, back into its chromosomes, and you'll also see that these chromosomes have got the 2 chromatids. Okay, so here's a couple of questions just to see if we understand. First of all, how many chromosomes do you see in the nucleus of this cell? Be very careful. Have a wee think. So the answer is that in this cell, right now, there are 1, 2, 3, 4 chromosomes. Okay, there are 4 chromosomes. Each of those chromosomes is made up of 2 chromatids. Okay, so it's very important we understand the distinction between a chromosome and a chromatid. Okay, this is a chromosome and it is made up of 2 chromatids. So in this cell, there are 4 chromosomes. Next question, how many chromosomes would be in a human cell? So you should know the answer to this because we've gone through that quite a bit. There would be 46 chromosomes if this was a human cell. Now, the reason why we haven't drawn all 46 chromosomes into this is it would just be a wee bit too difficult for us to see what's going on. So we've simplified it, and this, we can imagine that this is a cell that comes from a species where the chromosome complement is 4. Okay? All right. The next stage, stage 3, what we need is we need to have our chromosomes in a slightly more tidy format than they are even here. And we need them to line up along the middle of the nucleus. We call the middle of the nucleus the equator. So if we think of this as almost being like planet Earth, all the chromosomes are lining up across the middle, across the equator. And that would mean that the tops of this nucleus are called the poles. So imagine that's the north and the south pole, okay? So the chromosomes line up along the equator, and from the poles of the nucleus, the cell forms these special, they're almost, imagine it's like like ropes or grappling hooks that are firing out, and they attach onto the chromosomes. And in the middle of the chromosome, joining the two chromatids, there's a little part called a centromere. And that's the circles you can see there. So basically, the spindle fibers have shot out from the north and south pole, and they are joining onto the centromere in the middle of these chromosomes. Okay, so that is our third stage. In the fourth stage, so you can see this is stage 3 here. This is our chromosomes starting to move into the equator, and that's them lined up perfectly there. Okay, so in the fourth stage, what's going to happen is we are going to pull our chromatids apart. Okay, so the chromosomes that were made up of 2 chromatids were lined up along the equator like that. And in the fourth stage, the spindle fibers pull the chromatids apart to the opposite poles of the cell. Okay. Now we can see that going on here. So this is just another diagram, just another way of representing the same thing. So that was our chromosomes lined up at the equator, and this is the chromatids then being pulled apart to the opposite poles. All right. In stage 5, what we now see is that these chromatids from the original chromosomes have been pulled completely apart. And what then starts to happen is a new nucleus forms around that collection of chromatids. Okay. We would now refer to these chromatids as chromosomes, but they would be chromosomes made up of only one chromatid. The cytoplasm starts to divide, it pinches in the middle. And then the next step is that we are going to have 2 daughter cells. And inside each of these daughter cells, there is a nucleus. And inside that nucleus, there is 4 chromosomes made up of only one chromatid. Okay, so if we kind of go back to the start for a minute, okay, remember that when the cell was all tidy and organized and it had copied its chromosome, we've now got 4 chromosomes here made up of 2 chromatids. And at the end of the cell division process, we now have 4 chromosomes here, but they're made up of only one chromatid. Okay, and these 2 are identical daughter cells. So they have got the complete chromosome complement. Okay. Now, these cells, as they are, would not be able to divide. If they want to divide, they're going to have to do that DNA replication to add on a copy of each of these chromosomes, so that the chromosomes would be made up of 2 chromatids. Okay, just like we had back here. Okay. So that is the 6 stages of mitosis. Okay, the bit that some people sometimes find confusing is just really being clear on what we mean by a chromosome and a chromatid. Okay, so you do need to have a little think about that. And if you are unsure, do remember just to ask your teacher for a wee bit more clarification on it. Okay, so what you're going to need to do now is, at this point in your notes, you will have to try and complete your notes. Okay, I'll skip ahead to that for a minute, just come back. In your booklets, you should now be able to complete questions 1 to 8 on the producing new cells topic. Okay, that is subtopic 1. And you should then also be able to attempt the past paper questions on pages 4 to 6. Okay, so spend a bit of time. In one of the sections, you are going to have to number each of the stages of mitosis, like you can see here, and you're then going to have to explain what is going on in each of these stages. Use this video to help you with that, okay? Use the information that you can see here, so just rewind it if needs be and go back through it. But that is everything that you should need to get you started on producing new cells on the mitosis section. And there will be other work that goes with this. Okay, so if in doubt, just get in touch with your class teacher and they'll be able to give you a wee bit more information. But well done, you can now stop the video.